Hello and welcome to another edition of Capital Calculus. I am your host Anil Padmanabhan. I am in IIT-Madras today to explore its association with the most cutting edge of frontier science, mapping the human brain, and how this research recently published by the Sudha Gopalakrishnan Brain Center has given India a seat on the global high table for neuroscience research. Joining me in this conversation is Professor Mohana Shankaran Shivaprakasham. He is the head of the center and someone who's deeply invested in the intersection of engineering and health. Thank you for joining us, Professor. I want to begin with the Dharini, India's first population scale brain imaging repository. I believe it's the acronym for a 3D developing human brain atlas resource to advance neuroscience internationally. What is Dharini and what is the story behind it?
Okay, thanks for coming, visiting the center, and having us on this show. I'll answer the first question, then we can go to the background. Right? So, Dharini is today the world's most advanced 3D cell resolution brain image of the second trimester of the fetal brain, which is somewhere around 12 weeks to 24 weeks, right? Which is where the brain essentially, you can say, it's starting as a fetus and is almost developing into a child, right? So how we came about doing this project is something that I'll follow up in a couple of sentences. But what essentially this is, this gives you cell resolution. A cell is about 10 micron, right? About 1/5 to 1/10 of the human hair. And there are billions of cells, right, in a fetal brain. And there are hundreds of billions of cells in an adult brain. So when we started this project, our goal was quite, sort of ambitious or audacious, and actually many people said it's too lofty to sort of, accomplish in India. Work was started about 5 years ago. It was actually triggered by Mr. Chris Gopalakrishnan, our beloved and distinguished alumnus. Somewhere around 2015-16, after he finished his term at Infosys, he sort of was interacting with us, saying that can we do something in brain research at IIT-Madras, and that's been one of his passions. And what can we do, right? That will be at the frontier, that will be cutting edge, that will be truly advancing the field, but at the same time that's something that takes advantage of what is available in India in terms of skills, capabilities, the ecosystem. And it's interestingly, we didn't jump right into defining a project. We sort of had this as a natural discussion and sort of discovery, I would say. And what we realized at that time, people were doing mouse brain mapping. Right? A mouse brain is about 1 centimeter cube, just to give you a sense of it. Right? And why is this brain mapping? Right? And as practically all of us know, the brain is practically inscrutable today, right? At every level. Right? When we say we don't understand this person, actually what we are really saying is, we don't understand the person's brain, both real time and at a long term. Right? So there are two aspects to it when we say we want to understand the brain. Right? There is a structural aspect, right? What we call the form. Right? Then there is a functional aspect. Right? And in biology we always say form determines function. Largely it's true for most of the thing. But even at a simplistic level, if you want to understand something, the first question is, what is it? Right? And then what does it do? How does it do it? And why does it do it, and why is it that way? So if you take cardiology or heart, right? So we have seen the heart in extreme detail, and that's why you can see how much advancement has happened in cardiology. But if you take the brain, right, we have not seen the brain, the whole brain, and the reason for the brain, the reason why the brain is complex to see, because it's inside the skull for because of evolution, we want to protect the brain. So there's no way to see the brain in detail beyond a millimeter resolution. Take an MRI, you'll get a millimeter. A millimeter is too coarse, or I should say too big, to see the brain in any great detail, right? Because these cells we are talking about are in 10 micron, 20 micron, which means we got to image the brain at about a micron. Then you will be able to see the cell, and there are hundreds of billions of cells. And unlike other organs, the brain is also, we know the brain is complex because it is networked. It's not the 100 billion neurons that actually are the only power, it's the interconnection between them. And that's what gives us this functionality, right? I'm talking to you, I'm also paying attention to whether the camera is not falling, I'm aware of what is happening. How is this all happening, right? So there is a big interplay. To get to that far, one of the key things is to actually see what is inside, right? Which are the types of cells, how are these cells connected, right? So this is a long-standing, scientific endeavor which people want to do. So when we looked at this problem, why are people doing mouse? It turns out to do human brains, right? I'll give you some numbers. Right? If you were to resolve the human brain at a micron, the entire brain, you will end up with several petabytes of data, right? Not terabytes, petabytes. Right? So to give you a sense of petabyte, about 1,000 laptops, that's one way to look at a petabyte. Another easier way for you to connect or visualize is if you take the entire Google Earth, right, the Google Maps, as it was commercially called, every street, every corner, everything, it will be in the order of several petabytes.
This is the size of the human brain.
That is the size of the human brain if I were to resolve it at even 1 micron. And 1 micron is, it's not a very, very high resolution, I will say. It is a cell level resolution. We're not talking electron microscope. So now you can walk back. Now how will you end up doing this in the first place? So it turns out it is a big technology gap, right? So we said, so here we have a scientific problem, right, which will lead to medical applications if we are able to do it. But to reach that scientific goal, you need to develop technology, right, which can take a human brain. Of course, it has to be post-mortem, immediately after the death, as soon as possible, so that the architecture and everything is preserved. And then now you need to somehow image the entire brain at 1 micron, right? This entire 1.5 liters of human brain. So you can imagine, I'll let you run in your head, how will you go about doing this, right? So the way it is done is essentially you have to cut the brain in very, very thin slices, right? About the size of a cell, you have to cut about 10 to 20 micron. Then you have to image each of this section. There are about 8,000 to 10,000 sections. You have to image each section about 1 micron, right? And you now have to put it all back together digitally. Then you have a 3D reconstructed brain, right? Now this is essentially a huge technology endeavor. So we said we will take up this challenge, and as it has now been written and by many people, this was considered very difficult, very, very difficult. But we said we will take up this challenge. Then the initial support for this came from the Principal Scientific Advisor's Office of Government of India and Chris Gopalakrishnan, and we started building this platform. And there was a twist in the story. The project started literally 2 weeks before the lockdown in 2020. So this entire technology was built during the COVID period. Right? It's one of the well-kept secrets of the center itself. Now it has scaled to a level where the Dharini is in one way the first public announcement and first public dissemination of this data. One of the things we were very, very absolutely determined to do is we want to make this data globally public. Right? We didn't want to hoard this data because we are doing this to advance science, right? And science is for the humanity. And the first set of data, which is what Dharini is, it is released publicly. You don't even need a login. You can actually go to that particular website. You can actually see 3D cell resolution view of how a second trimester, which is a very important period in brain development in humans, starting from 12 weeks to 24 weeks, it's available fully public. It's obviously a global project. People from almost 5-6 different countries are collaborating with us, this center, and many of them actually spend time here at the center. And that is what has led to this thing. And today, as a result of all this, it has put India on the global map of this frontier area of human brain mapping.
We'll just come back to that in a minute, but I want to ask you a very basic question. Is this the first time that the human brain has been mapped? It's a fetus, but it's the first time?
Yeah. So let me get one level detail. What you're seeing is this technology platform. It is the first time such a large volume and a throughput can be obtained. So people have attempted human brain mapping, but usually it is selective regions. You take certain regions of the brain and you image that, and you create a map of that. The challenge has always been the whole brain, because the whole brain is where gives you the clues of how different regions are connected, how different regions are relatively positioned in terms of not just in terms of geography, cell types, everything. So to do that, you cannot do this at scale unless you have a technology platform. So it was the technology platform was the one that was missing, and today what you have is actually the world leading platform in terms of doing whole brain human mapping at this scale and at this throughput. And of course, as you rightly put it, we started with the fetus. Currently, the type of brains that are being done at the center range from fetus all the way to a very old age. In fact, the center is developing maps of the human lifespan, starting from early fetus all the way to old age. Along the way, various disease conditions, stroke, infections, developmental disorders, which are many of which are visible at even at a fetal level, at early child development disorders, then mid-age conditions that you end up having like multiple sclerosis, and aging, neurodegenerations, Alzheimer's, Parkinson's, various psychiatric disorders, and natural aging and neurodegeneration. So we are creating for the first time maps of different, and you can already imagine how valuable this will be, not just as a scientific resource for us to understand, because we don't practically know nothing about the human brain in terms of how it's organized. It will also give clues to clinical researchers and to clinical practitioners, because currently what they have is the MRI. That's all they have. They really can't see in detail of how this particular diseased brain is in various regions of interest that are relevant to medicine.
So quick follow-up. So just like you published the data for the fetus brain, you have not yet published for the other stages.
It's happening literally.
So in this year,
it is in this year we are going to be releasing some adult human brain related data resources.
Like a similar 3D mapping of the brain.
That's right.
So another basic question before I just take a deeper dive is that is imaging a dead human brain very useful, or is the only option?
That's a good question, right? So you actually framed it very nicely, right? Is it the only option? The answer is yes. There is no way we can do whole human brain, right? I can probably, in a diseased brain, take pieces of the brain tissue. Right? When I say pieces, I'm talking about millimeter cube. Right? In tumors, we actually take out few centimeters square to few centimeter cube. But if you want to do the whole human brain, you have to take out the brain, because as you know, light cannot penetrate. So if you want to do microscopy, right, 1 micron means you need microscopic level imaging. Light cannot penetrate the brain. You can do MRI, which is what is being done. It's about a millimeter. You can push it to about half a millimeter. But if you want to do micron level, 1 micron, actually we do half a micron, right? You need the brain, right? And that is actually, that makes the project more challenging, because now you need post-mortem human brains, right? So now you asked a question, right? You use the word dead brain, right? When you say it's a dead brain,
Okay.
is it useful? No, I mean it's the good way you framed it, right? Is it still intact? That's the first question, essentially. I guess that's where you were sort of alluding to. It is, if you extract, or what do you call it, acquire this brain in a certain high quality manner after the death within a certain time frame, right? Within a certain.
To do with harvesting other human
That's right. Of course, harvesting is generally, when we say we harvest organs, right? Generally, it is from, when the patient is, when the subject is still alive, right? Here it is done after the death, right? It is done after the death. So we have several collaborators in the country, leading medical institutions across the country actually, who work with us, who acquire these brains, right, in a very high quality manner. So what is quality? That's when we ask them. The quality essentially means you preserve the architecture, right? To preserve the architecture of the brain at a cellular level, at a connectivity level. And you all, because the brain has a lot of blood in it, you try to acquire the brain without too many clots. I'm getting into the secondary level details, right? So like this, we have acquired close to about 300 odd brains, right? And there is a pipeline from different institutions. So this itself was a project, right, by itself. And because of that, today we have a variety of brains across ages, across disease groups. And to answer your second question, to reinforce, yes, that's the only way, right? But for it to be accurate as a representation of what is in a live brain, you have to acquire it in a certain high quality manner, and we have actually developed that process. In fact, that process was actually published in American Journal of Forensic Medicine, which was sort of well received. You have made this thing public. Normally people actually don't make this kind of things public, but we have made this public.
You made a great point about how there's a pattern when India does such cutting edge research, whether it's digital public goods, now in the case of mapping the human brain, it's always doing it with a global good intention. It's never purely for monetary intent. So now I wanted to follow up and ask you that in a database of the scale that you have just drawn up for us, what kind of questions can researchers now, ask or rather answer, which weren't possible earlier?
Sure.
So, are there any specific neurological disorders or developmental areas this will help us? Because I'm asking in the context of the fact that India is now seeing a very big pivot in its health disease matrix. It's moved from communicable diseases to non-communicable diseases, and a lot of it is related to neurological factors.
So in fact, you actually framed it very nicely. Ask and answer, right? So you will be surprised that even what appears like a simple question, right, which is, how does the hippocampus, this is an important region for memory and other functions of the brain, how is the hippocampus of a brain with, let's say, Alzheimer's or neurodegeneration in a broader sense, how is it different, right, at a detailed level compared to same age group brain that doesn't have it? This question is still not answered, right? This is a very first order question, which is number of cells, cell types, right? Same with stroke, right? In fact, we realized when we did the stroke work, stroke work is actually supported by our alumnus, Mr. Prem Vatsa of Fairfax. When a stroke happens, right, there are regions around the stroke which are not completely damaged, right, that region is of great interest. Right?
Why it was not damaged?
Not fully damaged, but there is a damage. Right? So that area is of great interest. Right? Can we heal it? Through what means? And for you to answer that question, you need to know what is in that region. Right? Not at a higher level. Right? You got to know at a cellular level, which is about 10 microns, as I said. And what are the cell types? And what is the extent of the damage? Right? These are the questions that are being answered. Right? Some of the questions were already existing, some of the questions are being answered. I give you an example in neurodegeneration, I give you an example in stroke. In neurodevelopmental disorders at an early stage, even at a fetal level, when a particular brain has a developmental disorder, right, the only way for you to even before you look at cure, how is this developmental disorder manifesting at the cellular level in detail? That's actually both a neuroscience and a medical question. Right? So these are questions which some of the questions were already standing questions which are being answered, and now because you now have a whole brain at a 3D, right? So if I give you the atlas of the brain, actually there used to be an atlas there, that's literally the entire second trimester atlas. You can actually look through and then you can say, oh, I didn't realize this particular region has grown significantly from X weeks to X plus N weeks. Right? So that generates new questions. Right?
So what you're telling us is that to get a solution, you need to be able to ask the right question. To ask the right question, you need to know what you're looking at,
That's right.
which is what this atlas is all about. Oh, before I wrap up, one final question to you. Do you believe, the projects like Dharini and what you're undertaking in the center are starting to reposition India from being a user of neuroscience tech to being a contributor and a leader in this case?
So if you look at, as we talked about earlier, we are second to now when it comes to talent, right, globally. But when it comes to generating scientific knowledge, right, and frontier areas, we could have done better. Right? Many of these projects, you go to any project all around the world, there are Indians there. Right? But such projects should also happen inside the country. Right? And this is not just about geography or being nationalistic, but it actually involves more people when you're doing it in the country. Right? And we mentioned before, if it's happening out of India because of our larger cultural makeup, we invariably make it for the global good. So that's the first thing that does it. The second aspect of it, when you start generating scientific knowledge at an early stage, right? As we always say, today's science leads to tomorrow's technology. Right? Generally, it's been that way. Right? You take genomics. So it starts with scientific unraveling. Then the tools that you make to do that unraveling eventually becomes technology. Right? So when you start doing this kind of scientific projects, you inevitably become a technology leader in that area. Right? And that's also happening, right, in this project. Right? So it's got not only at a global level impact of making such data accessible to a larger portion, it also helps the country to become a technology leader in this area. And that's what we are actually doing, and we are very confident that over the next 10 years or so, we will be a global leader. And we have started, so this journey will continue over the next 10 to and beyond number of years.
Thank you, Professor, for this very, very insightful and informative conversation.
Thank you, Anil.
So, you heard the professor. Our ancestors mapped the world to create what we know as geography of the world today, to exploit its potential. Now, a group of scientists are doing very much the same thing regarding the brain. And as the professor laid out before us, this is going to create equally immense opportunities as we have done by tapping geography. Thank you for watching. Don't forget to subscribe to stratnewsglobal.tech on YouTube. Hit the bell icon so that you don't miss any updates. And please do share your thoughts, ideas and suggestions with us. I'm available on Twitter at Capital Calculus. I'll be back next week with another episode. Till then, stay safe.